Hi there. So in this video, we're going to talk about a cancer immunotherapy called CAR T cell therapy. So the immune system is very powerful. It can kill virally infected cells. It can kill cancer cells. But the immune system isn't perfect, and scientists have figured out how to genetically modify a person's immune system to hunt down and kill and destroy cancer cells. And that's what we're going to talk about today in this video called that covers CAR T cell therapy. So it's a fairly new therapy, but it's very powerful. So let's talk about this. First, we have to remind ourselves, how do T cells recognize a target cell? So if you look at this drawing here, here's a regular old CD8 positive naive T cell. And we know that T cells recognize their target cells using the T cell receptor. And then the T cell receptor, we have this whole complex in the T cell receptor complex. We've got the alpha and the beta chain that recognize peptides presented on MHC class 1 molecules. We've got the CD3 proteins and the zeta proteins that have ITAMs that can become phosphorylated and send a signal into a cell in order to activate. We have a CD28 protein on the surface of T cells that need to interact with the B7 protein on an antigen presenting cell in order to activate a naive cell. So that's how a normal T cell works. Unfortunately, normal T cells are not always the best at killing cancer cells, right? Cancer cells have figured out ways to evade the immune response. So scientists have tried to genetically figure out a way to convince T cells to recognize cancer cells through a very ingenious method that we're going to talk about in this video. So for us to understand how CAR T cells work, we first have to introduce the concept that cancer cells might have some protein on their surface that would make them an attractive target for an immunotherapy, right? So some molecule on the surface of a cell that scientists can target, that can direct an immune system attack toward. So first we have to think about there are proteins on the surface of cancer cells that can be recognized by proteins, other proteins. Now, in the past, we've talked about monoclonal antibodies being used as therapies. And those are very fine therapies. People get infused with monoclonal antibodies, and these antibodies have antigen binding sites that might recognize molecules on the surface of the cells. And this is a common therapy given to people that will direct a humoral immune response toward a cell, right? And that could be complement-mediated lysis, it could be opsonization, all sorts of things that antibodies can do that can bring upon cells. But in this instance, we're trying to get the immune system, specifically the arm of the cell-mediated immune response, T cells, to bring that upon cancer cells. So how can we direct T cells to kill cancer cells? We already know we can infuse somebody with antibodies to target molecules on a cancer cell, but we want to target the cancer cells with T cell response. And for us to talk about this new CAR T cell therapy, we have to recall something about antibodies, because we're going to use the power of antibodies to recognize a cancer cell. So if you recall antibody structure, antibodies are made of 4 protein chains, 2 heavy chain proteins, 2 light chain proteins. And an antibody has antigen binding sites at their tips, right? And these antigen binding sites are created by the variable region of the heavy chain protein and the light chain protein. Okay, so that's just a refresher of how antibodies work and how they can recognize pathogens, antigens, right? So remember the structure of an antibody, because that's going to be important here. So here we've got a cancer cell, and there's some protein on the surface of the cancer cell that we would love to direct T cells to recognize. So scientists have figured out how to genetically engineer a new receptor protein on the surface of T cells called a chimeric antigen receptor protein. So what does that mean, right? So receptors are just proteins that are found on the surface of cells. An antigen receptor is going to be a a protein that's going to bind to an antigen. So there's an antigen binding site on these proteins. And these antigen binding sites are going to recognize their target, their antigen, and in this case, the antigen's on the surface of the cancer cell. Okay, so these receptors are going to have an antigen binding site. Now, what else is this receptor going to have? It's going to have the ability to activate a T cell. Because if we want the T cell to attack this cancer cell, right, we really need two things. The T cell has to recognize the cancer cell, and the T cell has to activate. And in this case, if we're talking about a CD8 T cell, when a CD8 T cell activates, it will recognize cells and kill those cells. Exactly what we want in cancer immunotherapy. So we're going to use the power of antibodies to genetically engineer a protein that's going to recognize cancer cells. And again, recall the antigen binding site of an antibody incorporates the heavy chain and the light chain proteins of the variable regions. Okay, so remember that. So here's our cancer cell, and here's our T cell. So let's actually get into what is the chimeric antigen receptor that scientists have genetically engineered. So this is a man-made protein, if you will. So this protein, the first part of it, we're going to talk about the extracellular part that's involved in recognizing the tumor antigen, is going to be genetically engineered from an antibody, specifically the light chain variable region and the heavy chain variable region. So as I was circling before in the previous videos, the antigen binding site of an antibody, we're going to take that and use that to create our chimeric antigen receptor. So here's the variable region of heavy chain and light chain, which we know creates an antigen binding site. And we scientists have taken that from an antibody and taken those two domains and linked them together. And so this region of this chimeric protein is called a single chain variable fragment, and it came from an antibody. So again, you know what an antibody structure is. This domain is just from an antibody. And sometimes you'll see it labeled as SCFV because it's a single chain, right? Normally, an antigen binding site of an antibody is made of a heavy chain and a light chain. They're separate proteins linked by disulfide bonds. In a CAR, in a chimeric antigen receptors, the heavy chain and the light chain protein are actually linked by an amino acid chain, by a protein chain. So it's a single chain, and it's the FV, the variable fragment of an antibody. So if you recall antibody structures, remember there's the FC region, the constant fragment, the FAB region, the antigen binding site region. So if you just take the variable region of an antibody, we call that the FV region, the variable fragment. So this chimeric antigen receptor is made from antibodies, specifically the heavy and the light chain, linked together. To that, we're going to link a transmembrane domain, right? If we want a receptor, receptors have to be embedded in the membrane, so we need a transmembrane region. So that's genetically engineered into this protein. And then we need an intracellular domain. So what intracellular domain are we going to put here? Well, we want this T cell to activate when this receptor binds its ligand, which is a cancer or protein antigen. So what kind of proteins are involved in activating T cells? So if you remember back to our T cell receptor complex, the zeta protein in the T cell receptor complex is one of the proteins that has ITAMs in it, and those ITAMs can become phosphorylated and send the signal into the T cell in order to activate. So the intracellular region of the chimeric antigen receptor will often have fragments of T cell proteins that are involved in T cell activation. So sometimes you'll find the zeta protein, sometimes you'll find CD28. That's another protein that's involved in T cell activation. So scientists have played a lot with the intracellular regions, trying different combinations of proteins involved in T cell activation and putting those regions into this chimeric antigen receptor. And these regions are going to help send the signal into the T cell in order to activate it. So what we have just created here is a chimera. And we keep using that term a lot. For those of you familiar with Greek mythology, right, the origin of the term chimera, a chimera is an animal in Greek mythology that had parts of all different types of other animals. So the chimera had, I believe it had the body of a lion and the head of a goat and wings of a dragon and parts of it, like a tail of a snake. So this chimera that scientists have created has protein fragments from an antibody, protein fragments from a transmembrane region of another protein, intracellular protein region from the zeta protein, from the CD28 protein, from other proteins. So it is a chimera of a bunch of other proteins. That's why we call it a chimeric antigen receptor, right? And it binds antigen. It's got an antigen binding site. So the goal of this chimeric antigen receptor, the CAR, is to bind to antigens on the surface of a target cell, in this case, a cancer cell. So here are a bunch of CARs. They all have antigen binding sites. Those antigen binding sites will bind a cancer cell antigen, and that's going to bring these CAR receptors together. And as we mentioned before, ITAMs, intracellular tyrosine activation motifs, which are crucial for sending signals into the T cell. These ITAMs are often found in these proteins that scientists have decided to include in the intracellular region of the CAR. So the zeta protein, the CD28 protein tend to have ITAMs in them. And when the T cell, when the CAR T cell recognizes its target using its CAR antigen receptor, these ITAMs will become phosphorylated, send a signal into this T cell, and this T cell is going to activate against that cancer cell. So this is a chimeric antigen receptor, CAR, and those are the structures you'll see in some CARs. So let's bring it to fruition. How is this going to actually help a cancer cell patient survive? So when individuals are treated with a CAR T cell therapy, the first thing scientists do, clinicians do, is they isolate T cells from patients. So this is an autologous T cell therapy. We take T cells out of a patient. Those T cells are sent into a lab, and we're talking about CD4 cells, CD8 cells. And in those T cells, clinicians put in a new gene. And this gene has the instructions to make these CARs. So this gene has DNA information from antibodies, DNA information from zeta protein, DNA information from, I should say, the gene that encodes the zeta protein, the gene that encodes the CD28, a gene that encodes the transmembrane domain. So we're genetically engineering T cells to express this brand new chimeric protein, the CAR. And then these cells, we can now call them CAR T cells because they carry this chimeric antigen receptor. And these cells are grown in the laboratory. This process takes maybe 2 to 3 weeks. And these cells are infused back into the patient. So a patient is donating their T cells. Those T cells are genetically engineered to express the CAR receptor, and those cells go back into the patient. So remember, these T cells are going to do what T cells do, which is try to find a matching antigen. And in this case, the CAR receptor is going to look for its antigen binding site that matches the antigen. So it should ignore a bunch of tissue that doesn't have the antigen on it. But once it finds the cells that do contain this antigen, and in this case, it's a cancer cell antigen, these T cells are going to attack that cancer cell. These T cells are going to activate. So again, we've got the antigen binding site of the CAR, and again, scientists have chosen an antigen binding site that will match the antigen found on this patient's tumors. And the T cells will have their ITAMs become phosphorylated, and these T cells will activate against this cancer cell. And if we're talking about CD8 T cells and CD4 T cells, what are their effector functions? Well, we know that CD8 T cells, once they activate, they will release porphyrin, which puts a hole in the target cell. They'll release granzyme, which will go into the target cell and kill the cell. Normally, CD8 cells are recognizing and killing virally infected cells. In this instance, they are killing cancer cells. So now we have CD8 cells, the CD8 CAR T cells, killing a patient's cancer cells. And these CD8 cells are also releasing cytokines as well. CD4 CAR T cells, they're going to recognize the cancer cells and they're going to release cytokines to provoke an immune response. So now we have the cell-mediated immune response against cancer cells, which is a really great thing because that will and can eliminate the patient's cancer. Now, one of the side effects of CAR T cell therapy is something called cytokine release syndrome or cytokine storm. If you think about all these brand new T cells that go into this patient, the CAR T cells, they're going to go and recognize and activate against these cancer cells. So there's a massive T cell activation in patients who have CAR T cell therapy. So they have a massive inflammatory response called cytokine release syndrome, also called a cytokine storm. There's a huge amount of inflammation that's going on in their body. Cancer cells are dying. There's a huge amount of debris and dead cells in their body. Other immune cells are activating. So there is sort of a real danger using this therapy that the immune response might be so great and actually might end up damaging some other organs and tissues. But if patients make it through the cytokine storm, then oftentimes these T cells will have eliminated the cancer cells. So this is a really powerful therapy. Again, very new, but also very powerful. So hopefully in this video, you've sort of appreciated the structure of the CAR, understanding where those regions came from and how they're used to recognize the pathogen. No, it's not a pathogen. It's an antigen from a cancer cell, and how those CARs help activate T cells.